Good morning, good afternoon, good evening, and even good night, wherever you are on the globe. It's my pleasure to welcome you all, presenters, attendees, and moderators, to this third day of the workshop organized by the ISBT Working Party Global Blood Safety on Stepwise Access to Save Plasma Proteins in resource-constrained countries. My name is Martin Smith. I'm chair of the Working Party. Yesterday, we had 376 unique views, and I would like to thank all attendees for the many questions that were received. We were not able to answer all the questions, but all the questions are saved, so the moderators, with the help of presenters and panelists, will be able to address them afterwards. Before we start, let me remind everyone about the opportunities for participation. The speakers' presentations have been pre-recorded. The discussions will take place live. During the presentations, you can ask your questions in the Q&A box on the bottom of your screen. Some devices, it's on the top. The questions will be posed anonymously. Your name will not be visible to the audience. Questions that you ask will be answered during the discussion session after the end of each session. During the discussion period, you should not use the raise hand button. You can use the chat box feature if you wish to interact with the rest of the attendees, but do not use the chat box to submit your questions for the speakers. Use the Q&A box for this. If you have a question that is specific to a speaker, please indicate that along with your question. As a reminder, there will be a panel discussion at the end of today's program. You can submit additional questions that you would like to address to the panel. It's now my pleasure to introduce the moderators for the first session of today. Their introduction will be very short, as the bios of the moderators can be found in the workshop handbook provided to all. The moderators are Dr. Micha Nuebling from Germany. He's the head of the division Major Policy Issue at Paul-Ehrlich Institute. The other moderator is Professor Thierry Burnouf from Taiwan, and he's the secretary of the Working Party for Global Blood Safety of the ISBT. Dr. Nuebling will now introduce the speakers and topics for today's session.
Yeah, thank you, Martin. Welcome to this session addressing scale up for domestic processing of plasma, technology options, equipment, and oversight. The program for this session is intended to identify how industry suppliers may support the practical implementation of scalable plasma processing technologies in low and middle income countries, in part using single-use equipment and by providing technical and regulatory assistance. To cover this objective, we will hear the experience from 3 industry suppliers: Sartorius, with a presentation given by Prity Bengani-Lutz, Merck Millipore by Josephine Cheng and Jian-Fei Shou, and finally Asahi Kasei Medical with a presentation by Tomoko Hongo-Hirasaki. After these, we will hear about the practical implementation of a process to manufacture SARS-CoV-2 immunoglobulins in India as an example for domestic production of immunoglobulins. Dr. Suma Ray of Intas Pharmaceuticals will give this presentation. We think to be of interest to hear then which lessons we could learn from the fractionation of antivenoms immunoglobulins from horse plasma in low and middle income countries, with a presentation by Dr. Mariangela Vargas from the Instituto Clodomiro Picado in Costa Rica. And finally, we will end this session with a talk by Mohamed Ghonim of QSE Consulting in Switzerland, echoing some presentations given yesterday and today on the regulatory considerations for authorization of plasma processing devices. We will now commence the recorded presentations.
morning, good afternoon, good evening, wherever you're joining us from. We're delighted to have you. And today I'm excited to be talking to you about the contribution of single-use technologies for processing of plasma components. I'm Prity Bengani-Lutz and an employee of Sartorius Biotech. Just a brief overview about what we do. Sartorius is a global leading partner of the biopharm industry and the research sector. We help biotech scientists and engineers like you across the entire globe to develop and manufacture therapeutics from ideation to production so that more people would have access to better healthcare. At Sartorius, we empower scientists and engineers to simplify and accelerate progress in life science and bioprocessing. We have over 400 plus protein-based therapeutics, with plasma being one of them. We are a global company with headquarters in Germany, but worldwide locations in Asia, Europe, and America. We have over 21 manufacturing sites and have expanded in key areas around the world to meet our customer demands. Today, we will first start with a discussion with a question: why do we need single-use technologies and facilities? Then go over a quick overview of Sartorius solution offerings and services before we go into the specifics for the different process steps. We will cover 2 chromatography examples and then talk a little bit about filtration, virus and sterile filtration, and then close with how Sartorius can help you implement these solutions. So the need for plasma and plasma-derived therapies is growing rapidly, as I'm sure you've been hearing in the last couple of days as well, right? It's especially for autoimmune inflammatory diseases, immunodeficiencies, coagulopathies, and then with COVID-19 and the recent massive use of plasma-based therapies, the demand has gone up even more. There's a global product shortage, and while a large quantity of plasma in resource-constrained countries such as yours is being going to waste due to lack of knowledge and lack of support. We know that a couple of things that inhibit the progress has been the cost of a traditional stainless steel industrial facility, lack of skilled manpower, and of course, technical and regulatory knowledge. So there's a great need for single-use solutions that can address these challenges and help bridge the gap in resource-constrained countries. So single-use solutions increase flexibility and efficiency, both for the process as well as the facility. There's a lower risk of contamination than reusable technologies. Also, there's no hardware cleaning required in between, which greatly reduces the time as well as the cleaning validation requirements. And finally, single-use facilities are smaller. They require less capital infrastructure cost, which is especially important in resource-constrained countries. You also get rapid changeover from one equipment to another, from one process to another, and at the same time, it greatly simplifies scale-up and process implementation. So here's just a quick overview of things that Sartorius offers. We have single-use solutions for various processing steps, and most importantly, we provide an array of services to simplify the implementation process. Our consultants can help you from step-by-step implementation to choosing the right technology for your process needs to advising on the design of a new facility. We provide a range of services, including but not limited to those mentioned on the right. Product and process training to get you up and running quickly, process development, design, viral clearance studies, scale-up studies, and then fluid management solutions, process validation, and so on. And on the left, you can see we show a selected offering of our solutions. The top row shows the lab scale and bottom row shows large scale. Chromatography is used for removal of contaminants. We offer 3 different options: resins, membrane-based, as well as monolithic columns with different chemistries. Virus filtration is used for removal of viruses and other bloodborne infectious organisms. We offer Virosart hollow fiber filters. Sterile filtration offered for sterilizing and removing bioburden, and we offer both pre-filters and filters. We also have solutions for ultrafiltration and diafiltration, which I won't have time to go into today. So let's start with, now we're going to look at two specific examples for chromatography. This one is resin-based. And here you can see we're talking about the challenges in plasma purification. And before we get to challenges, let's look at the process workflow. So here's a sample process flow. Again, processes vary depending on the country, depending on the company, and so on. Here's just a sample, an example. So it starts with fresh frozen plasma and then forming cryoprecipitate. And to create cryoprecipitate, fresh frozen plasma is thawed to 2 to 4 degrees, then centrifuged, and the precipitate is collected. This is followed by virus inactivation by solvent detergent treatment or low pH treatment, which is typically involving caprylic acid, and where now you have fractionated plasma. And next is chromatography for contaminant removal and purification. And then you get pure IVIG. So let's talk about the chromatography steps highlighted in the gray box. The common pain points or challenges include low yield, low purity, and costly removal. Promising approaches include a combination of cation exchange and anion exchange. And the benefits include you get high yield, low cost, a lot of time savings with no additional dilution or UFDF step that's required between ion exchange. So let's look at a data slide that how we were able to obtain basically successful isolation of high purity IgG using resins. We started with a cryopoor plasma that had been subjected to 5% caprylic acid treatment. And then we performed cation exchange and anion exchange steps. And as you can see here, we got an IgG recovery of about 92%, and the IgG purity was about 99.8%, which is pretty good. And it also shows that you were also able to maintain the different fractions of IgG. We can also use membrane adsorbers for their polishing and improving recovery. Now, the second example is about isolation of IgG from cone paste using monolithic columns. And so here is just an example where the process is sort of similar. And you can see that typically the purification is done using cone fraction 1 and 3 only, rather than all 3 fractions due to clogging issues. But using monolithic columns, you can use all 3 fractions, and because of that, you get higher yield. Monolithic columns have large pores, so there's convective flow, and you get a fast, high-capacity process. And here, another quick data slide. I don't have time to go into how monolithic columns work, the technology, but I thought this slide will give you an idea about what sort of benefits you can get and what the capabilities are. So for example, this is about a nice example of how IgG was isolated from cone paste. In this example, you can see here's the peak that all of the IgG was recovered. IgA and IgM was also collected separately for further processing. You can see the table on the right shows that the IgA was completely removed, went from 5,000 milligram per liter to 3, and 70% of the IgM activity was preserved, as well as there was excellent aggregate removal. And this is something I'd like to mention too, that this is a very cost-effective process, as you can use the columns for at least 200 cycles with really robust performance. And again, if you want to learn more, please reach out to us. Now I'd like to spend a little bit of time talking about filtration. So like all other human medicinal products, plasma-derived medicinal products require a virus clearance strategy. These are especially challenging feed streams due to being extremely hydrophobic, high viscosity, and higher protein concentrations, which can lead to filter clogging, and it's just, yeah. So with that, Sartorius has a dedicated filter that's designed especially for plasma processing. It's the VirusArt HC. It has a retentive pore structure, and that basically enables the antibodies to go through, but the viruses are retained, and it results in high recovery. You get excellent filtration results and high throughput. In addition, it's a scalable technology. We have for small scale process development, there are filters available as small as 5 centimeter square area. So this way you only need small samples. These are fully scalable to pilot and process scale, and we can help you in developing a robust process. Now let's talk about sterile filtration. So sterile filtration is another area that's challenging because of the reasons we just mentioned above. And so, and one of the things is if the binding of the product to the filter leads to low yield typically. So we offer low binding pre-filters and filters, the SartoClean and the SartoBron P, and these offer high product yield. They give you this ultra low unspecific absorption. You know, they're really useful for large shear stress sensitive proteins. You get good throughput, good flux, especially for blood and plasma products. And we have all, transfer sets, filter transfer sets, and things like that, all accessories that you would need to integrate it easily into your single-use assembly. Now, in the next two slides, I would just like to emphasize how important it is that to partner with a knowledgeable company that really has the knowledge and can provide you the support along the way, because we know it's not that easy. And as you're getting started on the stepwise process, we believe that Sartorius really has the knowledge and the resources to help you along the way. We can help you in many different ways. We provide validation services, including extractables and leachables, bacterial challenge test, product integrity testing, viral clearance studies. And not only that, we also provide application and value chain services. We have a team of experts that are very knowledgeable in the process and can really guide you from process development to application support to process modeling and facility design. And we also show that our team of experts are available to you for many different options. We have product specialists that can provide application guides, application notes, validation guides, manuals, and so on for training and getting you up to speed. We have field application specialists that can provide on-site support for installation, training, and validation services. And then we also have process consultants or engineers for discussion of facility design, room classification, room segregation, what would be the total cost of ownership, making a GMP facility, and so on. So yeah, if you would like any further information about any of these things that I've talked about, any of the products, the solutions, the services, or how to get or talk about the specific needs for your individual process, please contact your local sales rep for Sartorius for more details. Or you can contact me too if you have any questions. Again, my email address is just my name at sartorius.com. So it's Prity.Bengani-Lutz at sartorius.com. I have the website there, the main website, and also the one dedicated to blood and plasma, so you can read more. And again, thank you for your time, and I look forward to taking some questions that you have.
Hello everyone, this is Josephine Cheng and Jian-Fei Shou from Merck. Thanks for the kind invitation of ISBT for providing us the opportunity to share with you the topic, exploring possible approaches for domestic scalable plasma processing. For content, we would like to discuss how to implement a stepwise approach for domestic plasma production, utilizing key figures from our public-private collaboration and our long-time industrial expertise in biological manufacturing. First, let's take a look at the current market situation. Even though the overall plasma market is prosperous, we unfortunately still hear a lot of shortage being reported from FDA, especially low and middle income countries continue to struggle from unbalanced accessibility, where access of quality plasma can still be an issue, and wastage of plasma resulting from this continue to be seen. Therefore, how to implement pragmatic and scalable technologies to make safe products to treat patients in need is an urgent topic to address. And that leads to our objectives of the presentation today. We want to explore possible approaches to make safe plasma and avoid wastage in resource-constrained regions. By this, we propose to discuss in 3 buckets to look at how novel technologies can help manufacturing plasma safely and efficiently, how to design a linear scalable process to ensure transition from small to large scale, and how to utilize the trend of single-use technology to foster the establishment of plasma purification capability. Now let's discuss how novel technologies can help increase purification efficiency. Here we would like to present a case study from our collaboration work with Professor Burnouf at Taipei Medical University, where the latest technologies were incorporated, aiming to develop a generic, easy-to-operate flow-through mode purification process with results meeting safety and purity requirements. The results of the work is being published in the journal and technical documents from our company. The summary of the major achievement is outlined here in the below diagram. We successfully used one-step anion exchange chromatography to reduce unwanted IgA and IgM. We used affinity chromatography to reduce blood type specific isoglutenins, and we used solvent detergent and reverse phase chromatography to remove the solvent detergent added in for the virus inactivation step. And all these chromatography steps were operated in the flow-through mode, so basically similar to the filtration process. In addition, we also implement the single-use TFF to achieve the final concentration of 20%, which is fit for subcutaneous IgG infusion. For the purpose of today's workshop, we will take parts of the process as some proposed solutions with experimental data generated in this study. First proposal, we would like to share with you the results of this one-step anion exchange chromatography using fructogel TMA in this case, how it greatly increased the purity of IgG just after one flow-through mode purification process step. As you can see in the bottom graphs, the unwanted IgA and IgM are greatly reduced, and the proportion of IgG subclasses have no significant change. The parameters and conditions were tested under 200 cycles, and therefore we are very confident with the purification result and the robustness of the process. The second proposal we want to look at the possibility to remove blood type specific antibodies, the anti-A and anti-B isoglutenins in this case, where patients can have reduced risk of hemolysis and manufacturers can have less work screening upfront with different blood type specific materials. Again, this affinity chromatography binds the unwanted isoglutenins, and therefore we simply need to flow the sample through the column and collect products at the end. The results showed significant reduction for anti-A and anti-B depending on the starting level, from 3 to 5 folds reduction, all meeting the regulatory requirement 1 to the 32. Proposal number 3 here, we add in a second step virus inactivation on top of the initial caprylic acid step, which is a classical solvent detergent method, and evaluated a flow-through mode reverse phase chromatography step to efficiently remove it. As we can see on table A here, Triton X100 with TMBP can effectively inactivate various model viruses in time as short as 5 minutes. And in table B here, we show effective removal of solvent detergent on the C18 column, leaving the sample with a low residual level almost below detection level. So those were 3 quick processes we proposed, and they should serve as a quick start to obtain plasma treatment in a stepwise manner. When the 4th capabilities are established in the region, we can easily connect them together and make a comprehensive process just like the big plasma manufacturers. Now, let's look at other aspects in plasma manufacturing, and I would like to hand this over to my colleague Jian-Fei Shou.
Thanks, Josephine. To establish domestic capability on plasma processing, we would have to have an understanding of cost structure. Think of designing a scalable process from the beginning to ensure smooth upscaling, and understanding how single-use technology can accelerate the setting up speed. Let's take a look for the cost analysis. The cost analysis was done using software modeling with data input using global sources, and therefore just serves as directionally correct to understand which unit operation would be costing the most in generic IgG purification process, and how upscaling can impact the overall cost per dose. So you can see the most expensive step lay in chromatography, virus filtration, and ultrafiltration. Therefore, optimizing these steps can make the greatest impact on overall cost reduction. As a comparison, the fully stainless steel process increased largely on labor cost, though there are some reductions in consumables and materials. So summarizing some observations through modeling cost analysis. Across the line, optimizing ion exchange chromatography, affinity chromatography, virus filtration, and ultrafiltration can make the greatest impact on both hybrid and traditional stainless steel models. The incorporation of single-use technology reduces a lot of capex and labor costs, mainly due to the elimination of operating large systems, cleaning, validation of all system, piping for each production run. The difference of cost per dose for the 2 models decreases when the scale increases. However, the incorporation of single-use can not only be more economic during small scale, require a small footprint, but also a lot easier to set up for facilities and employee training. So overall, single-use has many advantages, like easy to use, time saving, smaller footprint, and easy operation. And technology nowadays provide all kinds of products fit for different scale manufacturing. Here are some photos just to give you a better visualization of how single-use facilities comparing to traditional facilities, where a lot of stainless steel pipes and tanks and systems need to be maintained. Here is an example of vaccine case study in final fill process. The single-use process reduced nearly 70% of the time compared with the traditional process. On the bottom right, here also a single-use facility comparing to traditional facilities. The time need to construct, the overall capex and footprint, all are greatly reduced. So moving forward, a flexible single-use process is already a trend in vaccine industry. And this can also well apply for the plasma industry, especially when setting up a new facility in LMIC. Another key point when setting up the process is really to think scalable at the beginning to avoid unpleasant surprise when you scale up. A lot of time, benchtop R&D scales research doesn't make it to the large scale manufacturing, because at the small scale, we forgot to carefully select and design a process step or equipment that would be feasible for scale-up production. And this is why we emphasize the importance of scalability. To dive a bit deeper, the best is we could select the technology that can have linear scalability, which means no matter what scale, the performance remains the same. and there is no need to evaluate process parameters, or there is no unexpected performance at different scale production. Therefore, eliminate the time need for repeating evaluation on the process parameters and ensure speed to market. Now let's take a look to see if single-use devices also scale up well. This topic is worried by some people because sometimes the device looks different from traditional products. And the answer is yes. As you can see here, capsules are single-use, and the cassettes are multi-use TFF devices. In 3 different tests, they show comparable results for linear scalabilities, and performance is similar between single-use and multi-use.
Thank you, Jian-Fei. Now, how does a solution provider like our company can serve the plasma industry? We summarize it in 3 categories: products, services, collaboration, and training. We provide high-quality products and also high-quality dossiers to help our regulatory filing. We provide technical support, CDMO service, regulatory support, and we actively involved in regional associations and collaborations from academic and industrial level, providing technical trainings to the biopharm industry, and set up partnerships with key opinion leaders and strategic partners. To conclude, we hope this presentation can give you a glimpse of what possible solutions to be considered as we try to set up domestic plasma manufacturing capabilities. We hope to demonstrate that mature technologies are existing that can be well applied to make safe and efficient plasma products. And the stepwise approaches accommodating single use can foster the regional competency establishment. The products developed can start simple, such as SD-treated cryo, or more complex products like IgG, albumin, and factor VIII. The scales can be as small as 100 liters to start and expand as large as 2,000 liters later on. Last but not the least, collaboration is the key. Collaboration with technology providers can accelerate greatly the development time and help increase finally the accessibility in the region. We express special acknowledgement to both TMU team and Merck team. And with that, thank you very much for listening. We're open for questions.
I would like to thank you for giving me this opportunity to speak at ISBT Global Blood Safety Workshop. I'm Tomoko Hongo-Hirasaki, a lead expert of Bioprocess Division in Asahi Kasei Medical. After the launch of the world's first virus remover filter Planova in 1989, over 30 years history, nanofiltration has been recognized as a robust virus removal method for improving the virus safety of biological product and trusted use in manufacturing of plasma-derived product. Today, I'll talk about the potential contribution of nanofiltration to the virus safety of domestic plasma components, especially in LMIC. The main purpose of this presentation is to provide an understanding of nanofiltration, to reduce barriers to the use of nanofiltration in LMIC, then to facilitate the use of nanofiltration as a virus removal process. Here is the outline of my presentation. First, I'll explain key points of nanofiltration, and then the challenges for implementation of nanofiltration. Next will be a case study of implementation of nanofiltration. Finally, the conclusions. Let's start by explaining what nanofiltration is. These key points about nanofiltration are summarized here. Nanofiltration has been recognized as the most robust virus removal method by the plasma fractionation industry and in international regulatory guidance. It is applicable to various types of plasma product. It can remove various known and emerging balances based on size exclusion mechanism. Nanofiltration filters are available with different surface area, making excellent scalability and allowing straightforward scaling up for implementation from mini pools to large-scale batches of plasma fractions. Well, first point is, nanofiltration is recognized as robust ballast removal method. It is a requirement by global regulatory agencies to ensure the ballast safety of biotherapeutic product. Ballast inactivation and/or removal methods are necessary in production processes. Guidance document mentions that it is desirable to investigate the contribution of more than one production step for ballast reduction, and at least 2 orthogonal steps should be assessed. Also, nanofiltration is recognized as a robust ballast removal method in regulatory report, guidance document, and as established through over 20 years experience of plasma fractionation industry, as well described in this publication, as shown in this slide. And next, what are the main technical features of ballast removal filters? First, I'll explain more about the ballast removal filter called Planova. Planova is a single-use filter device, and this filter contains a bundle of hollow fiber membranes. The protein solution is introduced inside the hollow fiber structure and then gradually penetrate and flow through the fiber walls towards the outside. This hollow fiber wall has unique microporous membrane structure as explained in the next slide. This slide shows the pore structure of the membrane and the filtration mechanism. This photo is a negative image showing the pore structure of Planova hollow fiber membrane by electron microscopy. This large block is void. Narrow bridge between void is capillary. Void are connected three-dimensionally with several capillaries. This structure is repeated through the membrane thickness as shown in this drawing. Based on size exclusion mechanism, balances that are bigger than the pore size of the membrane are captured and therefore removed from the protein solution. Indeed, proteins that are smaller than the pore size of the membrane can pass through the pore without denaturation. This slide illustrates the relationship between the pore size of the nanofilter and the size of the targeted balances to be removed. This bar shows the size in nanometer. This filter targets the removal of medium and large size of balances, HCV, HBV, Zika, HIV, and G2 filters targeted the removal of even small size balances including power balances and HAV in addition to larger balances. Nanofiltration using Planova is largely predictable and was demonstrated to reliably remove various known and emerging enveloped or non-enveloped plasma-borne balances, including HIV, HBV, HCV, Zika balances, West Nile balances, Pavo balances based on size exclusion mechanism. From our long experience with working together with plasma fractionators, it was being published that Plasma filters are applicable to a wide range of plasma-derived protein molecules of various molecular weight, as shown here. Data also shows that filtration using Planova does not degrade plasma proteins. Excellent scalability. In this slide, with regard to scalability, numerous data has demonstrated that a virus clearance over 4 log is ensured consistency using a range of filter surface area, as shown here for small size virus model PP7. This right figure shows the linear increase in flow rate with filter sizes, evidencing the scalability of flux. We will now discuss the possible challenges faced when implementing nanofiltration in production process in LMIC. This slide summarizes the main challenges and proposed solution. Well, the main challenges that can be considered include how to use nanofiltration and how to optimize the nanofiltration to increase performance. Another is how to meet regulatory requirement and technological trend regarding virus safety. For these points, we can propose the following practical solutions. Hand-on training to use Planova filter. Providing in-depth technical advice and support services. Providing regulatory information and updated scientific knowledge sharing. On-site training on virus clearance study. The on-site technical support available is illustrated in this slide. For lab test, using small size filters, we assist in the protocol design, in preliminary filtration experiment, in training of operators, and in data analysis, and we propose optimal conditions. For virus clearance study, non-GLP tests using process solution are first done to check virus removal capability, and then GLP validation study can be done for regulatory approval. We also assist scale-up steps using larger size filters, pilot test, and commercial production. Therefore, at any stage, we do provide on-site support. Besides, we organize scientific knowledge sharing through our Planova workshop. This is an annual event that started from 1998 and gathered Planova users, regulatory KOL, to exchange technical information. It is important to note that Planova filters has been used in the manufacture of various biological products for almost 30 years worldwide, including LMIC. I will now introduce case studies showing the implementation of nanofiltration on immunoglobulins. This case shows the first IVIG product certified for virus safety by China authorities. This flowchart shows the optimization of filtration for virus validation study. From early process optimization using small size filter, and next process scale-up using medium size filter, and preliminary virus clearance study using small size filter, and then virus clearance validation study by Chinese authority for parvovirus removal studies. Each stage was supported step by step by our technical teams. This other case study is a feasibility study of nanofiltration on mini-pool capric acid purified IgG. This flowchart shows the preparation of capric acid IgG process. Mini-pool fresh frozen plasma was subjected to cryoprecipitation step, and then cryo-poor plasma was collected and subject to caprylic acid treatment. This caprylic acid treated IgG fraction was filtered on 0.2 micrometer and 0.1 micrometer filter, and then nanofiltration by Planova 35N for removal of medium and large size viruses and Planova 20N for further removal of small size viruses. Using small size filter 0.001 square meter, filtration pressure is 0.8 kg F per square centimeter. This figure shows the 2 hours filtration profiles for Planova 35N and for Planova 20N. For Planova 35N, 50 to 6 ml of CA-IgG could be readily nanofiltered on 0.01 square meter filter size. For Planova 20N, 16 ml of CA-IgG could be nanofiltered on 0.01 square meter filter size. Since caprylic acid is a virus inactivation step, for envelope viruses, nanofiltration of CA-IgG provides second virus reduction step for improving virus safety for both enveloped, non-enveloped viruses that can be considered when batch size increases. Finally, I conclude my presentation. Potential contribution of nanofiltration to virus safety of domestic plasma component in LMIC. One point is, implementation nanofiltration is a straightforward process from mini or small pools domestic plasma component to large-scale batches of domestic plasma fractions. Second point is technical support and experienced scientific knowledge sharing can be provided to LMIC to improve the virus safety of domestic plasma component. Thank you for your attention.
Good day all. Greetings from India. This is Suma Ray from Intas Pharmaceuticals. First of all, I would like to thank Dr. Thierry and his team for organizing such a large virtual event on plasma fractionation across the globe, which gives me an opportunity to present the work of my company, Intas Pharmaceuticals, and India, what we do in the world of plasma fractionation. Thank you very much. Now, as I said, I am an employee of Intas, which is headquartered in India, and I work here in the capacity of a senior vice president managing all operations of the plant. In this workshop, we have heard a lot about the plasma market globally, in US and in Asia. So I will give a brief introduction of the plasma market in India and the fractionators working in this country. This will be followed by a brief knowledge about the Indian regulatory scenario, and then introducing Intas Pharmaceuticals to this forum and our plasma fractionation plant, our journey, our success factors, our challenges, and our operational excellence. Finally, I shall end this talk with 2 case studies, one on the virus safety of immunoglobulins and the other on the COVID hyperimmunoglobulin. As we have heard about the plasma business globally, even in India, we have a plasma market of around 135 million USD with a CAGR of 11%. This shows that we are growing more rapidly compared to Asia or US. The plasma available for fractionation annually is 1.1 million liters, mainly from the blood banks in the country. All of this is recovered plasma from more than 3,000 blood banks, and also there are additionally imports of plasma as well as critical intermediate. The market primarily focuses around 4 products, the IVIG, albumin, factor 8 and then factor 9. India is moving more towards self-sufficiency in plasma products in comparison to past times. And in today's world, India has around 80 to 90% self-sufficiency in IVIG, 60% self-sufficiency in albumin and factor VIII, and around 90% self-sufficiency in factor IX. Amongst the top players of plasma fractionation in India are, of course, Intas Pharmaceuticals, followed by the Reliance Life Sciences. Both of us use cons fractionation for developing and manufacturing of plasma products. Verco and Himerus are two other companies that work on the chromatography platform, and the Bharat Serums is a fill and finish facility. There are several other newer players venturing into this business. The annual blood collection in India is around 11 million units, and of this 68% is componentized. There we see an improvement in the value chain from rapid testing to ELISA to chemiluminescence to the nucleic acid testing. And we see that the nucleic acid testing is growing year on year with 5% increase. Sorry, 500% increase. The regulatory structure in India comprises of the Central Drug Controls Organization, which is the central licensing authority, followed by the state licensing authorities. The National AIDS Control Organization is the one which makes the policy planning for blood safety program, the Indian Pharmacopoeia Commission that designs monographs and reference standards, and the National Institute of Biologicals, which does all the testing for the blood products. In India, if you follow the Drug and Cosmetics Act for the screening and testing of the transfusion transmitted viruses like hepatitis C, HIV, and hepatitis B, wherein we test the mini pool, the plasma pool, and the finished product. Now coming to Intas Pharmaceuticals, this is a leading integrated Indian pharma company with global operations. It is ranked as the 10th Indian pharmaceutical company and is also a dominant player in biosimilars and plasma therapeutics. Accord Healthcare is our global subsidiary with its presence in more than 85 countries in the world with a very strong pipeline for products. In addition to the pharma products, it has also commercialized 2 biosimilars in Europe, UK, Canada, and Brazil. This is the plasma fractionation unit of Intas, a front view, which is housed in Ahmedabad in India. The plasma fractionation plant, it is manufacturing plasma products since 2014, and currently we have a capacity of fractionating 300,000 liters of recovered plasma. But this does not happen in one day. We started off with an R&D scale of 2 liters, 5 liters, and 10 liters, followed by a pilot plant scale of 50, 100, and 250 liters. The commercial scales for each batch are 2,000 and 2,500 liters. The annual capacity started off with 75 KL, moving on to 300 KL, with gradually from 150, 200, and 250. The facility is licensed by the Office of the Drug Controllers General of India. And currently we have a state-of-the-art expansion ongoing for an addition 700,000 liters for fractionation of recovered plasma. Our success journey did not happen in one day. So we started off in 2005 when we collected the recovered plasma from India, sent to Korea for contract fractionation, and brought the finished product for marketing in India. This continued to 2014 when some of the products were developed indigenously in our own dedicated R&D. The fractionation plant that I just showed you was commissioned in India by the Drug Controller General's Office, and we received the commercial license for manufacturing of 2 products, IVIG 5% and 20%. This was just the beginning when in 2017 we developed the factor 9 600 IU and the IVIG 10%, both of which got commercial license, followed by factor 8 and low salt albumin in 2019. 2020, when the whole world was crippling, including India, under the COVID-19 pandemic, Intas Pharmaceuticals made a big headway by developing 4 products which were licensed for the first time in India by any Indian fractionator. They were the fibrinogen, PCC, IgG subcutaneous, and IgM enriched immunoglobulin. In addition, we also brought out factor VIII 500 IU and the extra purified immunoglobulin. The most important and major development was the development of the COVID hyperimmunoglobulin. We were one of the first ones globally to develop this product after Kamada in Israel. 2021 also, we have a huge array of products to be licensed, the C1 esterase, fibrin glue, VWF, Hep B hyperimmune, and the IVIG virus filter. Now, all these products have been developed in our in-house R&D facility with no support of tech transfer or any technology support from any other organization or company. Now, like the rest of the world, Intas's plasma business is also growing year on year. As you see, we are currently at 34 million USD with a CAGR of 30%. Likewise, for IVIG also, there is an increase in the production year on year with a 25% growth, including an increase in revenue also. The factors that contributed to our improved access to the plasma-derived products were a dedicated talent pool, a vertically integrated business model, a very focused R&D with a dynamic pipeline, sustainable commercial strategies with a social angle to serve patients first, and continuous investment, innovation, process improvement, and capacity expansion. This gives us a 1 million capacity by 2023. It was not just a bed of roses for us all the time. We had our share of challenges also, particularly the talent pool retaining and acquiring. Now, you know that plasma fractionation requires a very niche talent pool. Now, to acquire this kind of talent pool and to retain them when there is a lot of competition from the emerging fractionators is a major challenge. Then there is a lack of focus for creating a robust and sustained model for fractionation program by the policymakers. There is a limited pool due to voluntary blood donations and a lot of dependency on blood banks. The cost of imported plasma is very high. There is lack of awareness of plasma product manufacturing and its usage. The government price control on finished products is a major challenge because if we want to increase our capacities, improve our processes, include the state-of-the-art technologies, that increases our COGS and decreases our revenue due to this price control followed in India. Also, very importantly, there is lack of global credibility and acceptance of India as a fractionation hub, either for indigenous fractionation or contract fractionation. Now, coming to the product portfolio of Intas, we do have a lot of products in immunotherapy that includes the intravenous immunoglobulins of different strengths and the subcutaneous immunoglobulin, as well as the IgM enriched immunoglobulin. We have the albumin of different strengths, including the low salt albumin. The hematology products include the factor VIII, factor IX, fibrinogen, and PCC. In addition, we have a huge product pipeline with different products at multiple stages of development. Most importantly, the COVID hyperimmunoglobulin, which is in clinical trial phase 3. We have the C1 esterase, fibrin sealant, VWF, alpha-1 antitrypsin, antithrombin 3, and the different hyperimmunes, all waiting to be commercialized very shortly. Now, as I said in my earlier slide, that we are a vertically integrated business unit. This is what I want to explain here, that we have a controlled supply chain from plasma to patient. Now, if you look above this dotted blue line, all the activities here are done at the blood bank. So it is the blood bank that selects its donors and takes the donations as per the different regulatory guidelines. And the donations are then tested, screened for the different transfusion transmitted virus, and the negative units are then componentized. Now it is this recovered plasma that we collect as a fractionator. Now, first of all these blood banks that collect blood and convert it into plasma are actually audited by us. And most importantly, they are all licensed by the Drug Controller's Office in India. So once we collect this plasma, we transport it under cold chain to our licensed storage in our plant, where it is stored in the quarantine cold room till it is sampled and tested by our quality control laboratory. And only those units that test negative, both for ELISA and PCR, for hepatitis B, C, and HIV, are actually released for fractionation. Fractionation of the critical intermediates are followed by the downstream purification into different products, and finally the drug product is manufactured. The product is released as per the Indian pharmacopoeia, but if you see all along we have very stringent checkpoints wherein the any kind of contamination, if at all present due to the transfusion transmitted viruses, are checked by testing of the mini pools, the plasma pool, as well as the finished product. Also, we have dedicated orthogonal virus removal step unit operations, both in fractionation as well as downstream purification process, which are robust enough to take care of all the other emerging or unknown viruses that could be possibly present in the plasma. So the fractionation based platform that we use for our immunoglobulins are of two types. For immunoglobulin 5 and 10%, which is for intravenous administration, we have both maltose and glycine formulations. And virus safety is ensured with ethanol precipitation, solvent detergent treatment, low pH, and 20 nanometer virus filtration. Similarly, for subcutaneous immunoglobulin in glycine formulation, we have the solvent detergent and the 20 nanometer virus filtration. Now, this is a summary of the virus spiking studies that was done on the immunoglobulin preparations at a renowned virus laboratory. So we see that we have dedicated virus inactivation or removal technologies for adequate removal of both enveloped and non-enveloped viruses for our immunoglobulin preparations. This determines the safety of the product. Now, with this knowledge in our kitty, we decided to develop the SARS-CoV hyperimmunoglobulin as a potential treatment option for passive immunization for COVID-infected patients. And for this, we use the convalescent plasma from patients recovering from COVID-19. And this is again as per WHO guidelines. Now, we ensured that the product is of quality that meets the global standards. We ensured safety of the product from all transfusion transmitted viruses. We had 2 dedicated orthogonal virus clearance steps, and also the product was tested for toxicity, pyrogen, endotoxin, bioburden. For efficacy, we use the in vitro virus neutralization assay and immunological assays, which I will show more in detail in one of my following slides. Now here you see the comparative flowcharts of the IVIG intravenous immunoglobulins as well as for the COVID-HIG. So this process was primarily derived from the basic IVIG process, wherein we used both 2 different orthogonal virus clearance technologies as well as chromatography for purification of the product. The purity profile shows a monomer dimer of more than 99% and an IgA of around 0.03 mg per ml. Here we show the efficacy of the product when challenged with live virus assays, where we see that the titer is more than 1 in 640 for plaque assays PRNT90, For serum neutralization assay, it is between 1 in 2560 to 1 in 5120. With the immunological assays for the surrogate virus neutralization, we have an inhibition of around 97% and roughly between 245 to 455 AU per ml of SARS-CoV-2 antibodies, in fact, the neutralizing antibodies. We have done a clinical trial on this product, and the results show that the product was safe and well tolerated. There was a lot of benefit derived from the product, especially for high-risk group patients with diabetes in the test arm. There was a decrease in the CRP IL-6 D-dimer in the test arm, showing its immunomodulatory effects. Neutralizing antibodies for SARS-CoV-2 were higher in the test arm and stayed for a longer time. And the virus clearance by RT-PCR for COVID-19 virus showed a 2.5 median days earlier clearance in the test arm. Now, major challenges faced for developing this product were definitely it was developed during peak pandemic season when availability of manpower was very difficult, availability of convalescent plasma was a challenge, and getting a slot in the analytical virus testing laboratory was also a very difficult way, as well as credibility from the subject matter experts, because developing such a unique product in India definitely raised a lot of eyebrows. However, there was a lot of support from our suppliers, GE or Cytiva, both India and Sweden. I appreciate the efforts of Mr. Vagu and his team in India. And from Asahi India and Japan, would like to appreciate Mr. Dabas and his team here in India, both of them who took the fast track way to get these products available for us in this short time during the pandemic. So finally, the take-home message is that India is getting very close to self-sufficiency in plasma-derived medicinal products in order to meet the domestic market demand as well as to supply to other countries. There is a paradigm shift in blood banking quality and regulatory standards. There is continuous improvement with stringent controls from plasma collection to delivery of finished goods, and increased investment in plasma-derived products. So finally, my concluding words that India and definitely Intas is a potential hub for indigenous and contractual fractionation of cost-effective and quality PDMPs of global standards, be it from source plasma or recovered plasma. With that, I would like to thank all of you for your attention, and stay safe, and appreciate giving me an opportunity to present in this forum. Thank you very much.
Hola, my name is María Ángela Vargas Arroyo. I am a researcher at the Instituto Cleomiro Picado, located in Costa Rica. And first of all, thank you for the invitation to participate in this workshop. I am going to present you an analysis related to the plasma fractionation of animal-derived immunoglobulins in low and middle-income countries, specifically the case of Costa Rica, and particularly about the lessons to be learned for human plasma IgG production. First, I'm going to present you an overview of Instituto Cleomiro Picado from a historical perspective. Then I'm going to address how is the manufacture of antivenom immunoglobulins performed and its similarities with human IgG production. Then we are going to have an analysis of which are the key points or evidences that make Instituto Cleomiro Picado a reference manufacturer of biologicals in low and middle income countries, which has been the main challenges over the road and which should have been done differently for better and quicker success. Finally, I'm going to end with some recommendations that could be useful for the audience in order to have a domestic fractionation of plasma-derived medicinal products. Before talking about the beginnings of the Institute, it's really important to mention that snakebite envenoming is a highly relevant public health problem that affects the most impoverished populations worldwide. In this sense, it has been classified as a neglected tropical disease by the World Health Organization. And also it's important to mention that the parenteral administration of antivenom immunoglobulins is the only scientifically validated therapy for the treatment of these envenomations. And therefore, like in the case of human plasma-derived products, they are included in the whole list of essential medicines. So, aware of all these problems in Costa Rica, an important effort began at the beginning of the last century, at the moment led by the really famous scientist in Costa Rica named Clomiro and this effort involved during the years many people and ended with the founding of the ICP in 1970 after a collaborative program between the Ministry of Health, the University of Costa Rica, and the US Embassy. 2 years later, the ICP became part of University of Costa Rica, which is a public institution, meaning that the ICP is actually a public institution, an aspect that will be analyzed further in this presentation. It has been a journey of over 50 years since the first batch of 242 vials of antivenoms were produced in 1967. In each decade, important contributions were made by numerous people until the ICP became a reference center of the manufacture of antivenoms. The first decades can be considered as a consolidation stage not only of the production of antivenoms, but also of the development of two important activities within the university, that are the basic research and also the social outreach to the communities. This other period, little by little, small but significant technical innovations were introduced in the manufacturing of antivenoms, but a main focus was on the development of and the strengthening of the basic research, which also led to the generation of a dynamic network of cooperation with international groups and manufacturers. And then in this other period, there has been a strong focus on significantly improving the technological platform for the manufacture of antivenoms, the product diversification, the improvement of the overall procedures, and the exportation of antivenoms overseas. And nowadays, around 120,000 vials are produced per year. And more importantly, main growth indicator is that we have grown in staff. Now our group is composed of 83 employees, which is the main basis of any productive activity. Entering into more technical details, snake antivenoms are polyclonal immunoglobulin preparations that are obtained from the plasma of animals that were previously immunized with snake venoms. So the manufacturing process starts with the venom collection by milking usually snakes that are of medical relevance in the region where the antivenom is manufactured. Afterwards, there's a careful selection of healthy horse plasma donors, and after an immunization schedule with a venom or mixture of venoms, there is the collection, quality control, and storage of plasma bags until they are pulled and fractionated into antivenom immunoglobulins by dedicated purification steps to remove unwanted protein contaminants and provide viral safety. Afterwards, the antivenom is formulated and stabilized, and then there is the filling of the antivenom into final containers and the final step of quality control of the final products. So antivenom manufacture can be described as a cycle that is composed of different units, starting with the biotermin for the husbandry of lab animals for the quality control assessment of the products. Also, the research and development department for the continuous improvement of the activity. The serpentarium for the production of venom pools that are used for immunizing the horses, so the horses are maintained in a horse stable in order to collect hyperimmune plasma that is fractionated and transformed into antivenom vials in the fractionation facility, and also the quality control in order to control the quality in each of the different steps of the cycle. All of these under a culture of quality assurance by following good manufacturing practices, quality by design, and risk assessment and traceability. Also, all of these is in order to perform a proper distribution of the antivenoms where they are needed. When comparing antivenoms and human plasma IgG, There are several similarities among both products, starting with the fact that they both share a similar active ingredient, as well as all the regulatory framework, the fractionation facility and equipment. Also, the downstream processing technologies are really alike, as well as the personnel in charge of the different operations, all the strategies to avoid bioburden contamination, viral inactivation, being that they are biological products, all the product formulation and stabilization, and also the quality control of in-process and final products are really alike. When analyzing the differences among both products, probably we can find several of them. But however, I want to point out just two of them. One of them is that human plasma IgG are medicines that are used for different diseases, and they have a global reach. And in the particular case of antivenoms, they are regional products used for just a particular disease. And the aspect I want to point out the most is that human plasma IgG, in most of the cases, is produced by big pharmaceutical companies, in contrast to antivenoms that, since they belong to the neglected disease group, they are produced by small manufacturers in the majority of the cases. So we can find an analogy between antivenom producers and the producers of human plasma medicinal products in low and middle income countries. So this way, it's really important to do an in-depth analysis of ICP as an antivenom producer, so that all the lessons and the different aspects that have helped and experienced the ICP within the years hopefully will be helpful and contribute to plasma-derived manufacturers in low and middle income countries. So what are the key aspects that have contributed to ICP's success in becoming a reference center for biologics manufacturing in low and middle income countries? Well, one fundamental aspect is that, as I mentioned before, ICP is a public institution that belongs to a public university, and therefore it has permeated the university principles and its platform. Likewise, the ICP has been surrounded by a strongly academic environment which has nurtured its scientific activity. In addition, due to these characteristics, it is a non-profit organization with philosophical basis that promotes social responsibility. An interesting fact is that around 70% of antivenom manufacturers worldwide are public. A second aspect is the regional and global contributions in which ICP has been involved, because ICP has participated in dynamic scientific and commercial collaboration, which has led to produce antivenoms against snakes from other regions, technology transfer agreements, which have also given the institution financial and economical sustainability. In this same sense, the generation of cooperation and networks with different stakeholders, as well as public-private alliances to promote the distribution of antivenoms, has been transcendental. Finally, the research and development has catapulted the ICP into an era of innovation of technologies, products, and improvement in quality and diversification. It is so important, it is worth deepening in the analysis of the dynamic role of stakeholders. Actually, for many years, snakebite and venom lingered without much attention, but recently, unfortunately, there has been a concerted multi-component and multi-stakeholder approach. In the last years, there has been a growing global awareness of the impact of this tropical disease, thanks to a diverse set of action and actors, to a network of different synergies, and they have created a framework of solidarity, cooperation, and commercial interaction. So, for example, there has been a strong network that has been occurring between the manufacturing laboratories from Latin America, and they has established like informal and really dynamic intergroup of consultations for troubleshooting in antivenom production. Also, ICP, through different collaborations with academic, research, governmental groups, has contributed to solve all the antivenom crisis by producing new antivenoms for countries in other localities. As I mentioned before, another aspect that has been of paramount contribution for ICP is the strengthening of research and development in two ways. One is in the improvement of processes and products by improving the safety, productivity, efficacy of antivenoms in different manners, and also by the development of new products, either antivenoms towards different snakes in different countries, and also by the product diversification, which also has led us to enter in the human plasma fractionation. For doing this, and mainly based on the experience acquired within the years of fractionating echin plasma, we developed a new technology for human plasma fractionation based on the separation of plasma in an aqueous-based system and obtaining two enriched fractions, one enriched with IgG and the other with albumin, and adding subsequent downstream processing steps until purifying in a parallel way both medicinal products. This technology was already patented and granted in several countries and also licensed in 2017 to a company located in Colombia. And this initiative is currently under development in Costa Rica, so we are also interested in all the experiences that we can acquire in this workshop so that we can also apply it to our reality in Costa Rica. In the same sense, and as in most initiatives, ICP has faced challenges and limitations throughout its history, and some of them continue to be part of the reality of the institution. The first issue I want to analyze is the organizational. The advantages of being a public institution within a university also have the disadvantage that the university administrative structure has not favored manufacturing and technological development and has generated weak managerial practices. For example, historically, there has been limited estimation of production costs, purchasing and maintenance programs, administration of inventories, organization of the flow of activities, distribution and marketing of antivenoms, as some examples. Closely related is the economic factor. Since the manufacturing of biologicals is specialized and expensive, the ICP has been in a constant fight to maintain the financial sustainability and self-sufficiency. Coupled with the fact that being a neglected disease, there is limited funding for low and middle-income countries and snakebite treatment. The next aspect is the regulatory one. There have been meaningful difficulties in GMP compliance. In addition, as it most likely happens in other low and middle-income countries, national regulatory agencies lack of experience on how to regulate antivenoms, and so it has been necessary for the regulatory agency and the ICP to learn together in this process. The technical gap has always been a great challenge. For a long time, the staff prepared the procedures based on pioneering and artisanal work because there was a notorious lack of investment in infrastructure and equipment and in staff hiring and training. More recently, ICP has made significant investments in these matters, as well as in the training and upgrading of the qualification of its personnel. And the last aspect is that there has been a chronic deficit in antivenom availability in various regions of the world, most notably in sub-Saharan Africa, and also there are limited channels of distribution and intermediaries and elevated market variability. In addition to the fact that ICP is continuously exposed to the emergence of new competitors. Under this scenario, what should have been done differently for better and quicker success? Well, based on the learnings and all the experience of the ICP and applying this slide to initiative of starting the human plasma fractionation initiative. So first, that you have all the project management, funding, and quality assurance culture as a base of all the initiative. It's really important that you assure that there's actually a real need and a problem to be solved. In this way, you will have a guarantee that you will have users or buyers that will acquire your product once you have it ready for merchandising. Then, to establishing a plasma supply that is not only enough in quality but also in quantity is really, really important. For this, it's really important also to have at the beginning a feasibility study to assure that the whole initiative is profitable. In the case of our experience in human plasma fractionation, something that happened is that we did a feasibility study really late after the initiative started, and in that time we realized that we had not enough plasma so that once that we have the products, the initiative will be profitable. But once you establish all of these aspects, then you can go to the regulatory framework involving all the GMPs, the national and international guidelines. And based on this framework, you can start to design and to establish the fractionation facility and the equipment required, all the design of the procedures, design of the products, following all the regulatory requirements, hiring and training the staff, and also designing and establishing, validating the quality control of the in-process and final products. All of these happen at the same time, and really, really important in the way that you can go to register the product. First of all, it's really, really important to get first all the procedures and the products involved in these 4 areas validated, and then you can go for the registration and then the distribution of the products, focusing also in getting all the intermediaries, all the channels of distribution, and really, really important also establishing a culture of research and development so that you can enhance and assure the continuous improvement of all these processes. After the analysis, in here a series of recommendations that can be taken in account, such as promoting the manufacture of cost-effective products so that they can be supplied at affordable prices and in sufficient quantity, promoting constant scientifically sound efforts in research and development for improvement of product design and manufacture, and strengthening the national regulatory agencies, performing investment in infrastructure, equipment, staff, process automation, also promoting technology transfers, public-private partnerships, and international alliances, and final, the importance of enhancing the positioning and credibility of products manufacturing in low and middle-income countries so that they can be also seen and a guarantee of quality. As final take-home messages, I want to emphasize that the overall process experienced by ICP is a result of pioneering, visionary, and passionate effort of many persons for more than 15 years. So it is of paramount importance to firstly set the basis regarding to the mission, vision, and strategy of the group so that they maintain full commitment and will, focusing on the integral management of the human resources and strengthening their capacity of establishing networking and cooperation alliance. This way, we need to believe that there is full potential of low and middle income countries to find domestic solutions for improving access to safe and affordable plasma-derived medicinal products. It has been a pleasure to share with you in this workshop, and I am at your service in case you have any comments or doubt. Thank you very much.
Good day, everybody. Today we will discuss about the regulatory considerations for authorization of plasma processing device, specifically pathogen inactivation. My name is Mohammed Ghunaim. I'm the Regulatory Affairs Director of QSA Consulting. Prior coming across our topic today, I would like to declare the following. I have been a Regulatory Affairs Director for a blood bank company. I have supported a virus inactivation medical device manufacturer for CE approval. As a matter of fact, classical fractionation products are regulated under pharmaceutical legislation. Blood bank pathogen reduction technologies are available for single and pooled fresh frozen plasma, as well as single or pooled platelet concentration. Those blood bank pathogen inactivation technologies are regulated under medical device legislation. We will focus today on the pathogen inactivation devices. Pathogen inactivation devices available in the market are Sorolane, Riboflavin, Methylene Blue, TMBB Triton, and the caprylic acid. Because of the diversity of the audience, I will subdivide my training into 4 parts. One, starting with the manufacturer obligation, and then the rule of notified body, and then the rule of National Regulatory Authority, and finally the rule for blood bank. Manufacturer. Before placing the product in the market, the manufacturer has to comply with the medical device requirement by making available general safety requirement for the product, labeling requirement, risk management file, usability study, performance validation, biocompatibility, design control summary, clinical investigation if it has been required, clinical evaluation, pre-market surveillance, prospective and retrospective post-market clinical follow-up if it is required by the national regulatory authority or notified body, and then the manufacturer has to comply with quality management system requirement. Pathogen inactivation device available in the market right now are classified according to class 3, which is a very sophisticated and very long-term process. However, in this slide, I would like to clarify that there is another option available or another route available for the manufacturer, which will be illustrated in the next slide. From the regulatory, the European regulatory standpoint of view, clarified in the Medical Device Directive, the product can be approved either through Route A or Route B. Route A will be the manufacturer will approve the medical device or the bags containing the chemical compound together in one product. Then they will seek approval from limited number of notified body available for class 3 approval. Actually, in Europe, class 3 is the maximum class. In the United States, they have also class 4. But class 3 here is the most risky device. So the available notified bodies designated to approve these devices are limited. However, there is another option available for manufacturer or another route which is available in the Medical Device Directive since 1993, which called the procedure pack. And actually this product is likely to be approved like procedure pack because, you know, they can approve the medical device itself, which is the bag containing without having the chemical inside. And then they got the approval from notified body as like the blood bags, like channeling device or storage device. So it will be classified according to its intended purpose, like class 2A or class 2B. This process takes from 6 months to 9 months as maximum. And then, so somebody might ask, what about the chemical compound? Chemical compound can be approved from the National Regulatory Authority. So the National Regulatory Authority will accept this product like a class 1 medical device. But this device has to be clearly identified in the instruction for use that it is intended for virus inactivation. So finally, the approval comes from the National Regulatory Authority. Then the product can be freely sold in this market. I think we are ready now for a small journey into when the notified body receive the documentation from the manufacturer, how they handle it. They start with assessing the performance, safety, and the efficacy of the product by reviewing the documentation, the technical file of the document, and they have a specific expert in order to review everything. For example, if the device is sterile, so there is specific expertise at the notified body premises in order to review this. They have to have actually clinical expert in order to make sure that the product documentation available are compliant with the requirements. Then they ensure that the tests being conducted on the product are effectively conducted, including the capability of the lab conducting the testing. Then they decide to make audit on the manufacturer himself on their premises, or even they sometimes extend their audit into raw material or critical service provider. Like, for example, if the manufacturer making the sterilization outsourced, they have to go to the outsource supplier for sterilization in order to make sure that the sterilization is being conducted effectively. And sometimes they might decide to make an audit, unannounced audit, so they are not notifying the manufacturer before they come. And they have to do this every 5 years or along the 5 years of approval. Also, they might take the decision to audit the supplier of the raw material. This is why they request, the notified body request that the company supply them or make a short list of critical supplier, and the manufacturer has to have an agreement with the supplier in order to allow the notified body to go to the supplier premises, raw material supplier premises, in order to audit them as well. And if they have found a major or non-compliance, the non-compliance goes to the manufacturer himself, not for the raw material supplier. And then they keep monitoring this. For example, they monitor, they audit the 13485 every year, and they recertify the company every 3 years. And regarding the CE, they are doing every 5 years complete cycle of approval. Every year they go to the company, and then by the 5th year, they start to recertify the company for CE from scratch. And then if the company fulfill these requirements, so it is the company can take the certificate of approval from notified body. A national regulatory authority has to ensure the compliance for the performance, safety, and the efficacy. And they have a set of criteria for approval according to the risk benefit. However, I can classify actually the countries in the whole world. Either they have tightened the inspection, that they have pre-market control, so the product, before the product come into the market, in the very first year, they control and they collect all of the information to ensure that the product is safe. But after then, they start to relax the requirement and they leave the product, even if they have bad feedback about the product. However, some other countries, they have very relaxed control in the very beginning, but they make control on the product after performance. They leave the product to come to the market, and then after 1 year, 2 years, they check in the hospital how the product perform. I call it relaxed control or post-market control. From my point of view, I recommend the pragmatic control, which is a mix between this and this. I will describe this in the next slide. So some national authorities, following the tightened inspection in the world, they are asking to audit the facility. They make a big checklist for GMP compliance, and they go to the manufacturer facility in order to make sure that they are complying with this. They ask for approval from other national regulatory authority like FDA, and they ask for CE certification, and they ask thereafter for testing at national laboratory for 3 batches, and then they ask for technical file in order to review them. But for the post-market, they are doing just market surveillance every 2 years, every 3 years, and they wait until there is a vigilance case coming up to the manufacturer. Then they respond to the issue. So they are doing very overwhelming here, and they are doing very little here. Other countries, national authority, they are doing a relaxing inspection technique. So they are doing very simple checklist, and they see CE or FDA approval, and then thereafter they check, they follow up with the market surveillance, follow up with the blood bank, follow up the vigilance status of this manufacturer. However, our recommendation, my recommendation here is to follow the pragmatic inspection technique. So you have to detail the checklist in the very beginning, ask for free cell certificate if available, and CE and the FDA, and not to reinvent the wheel again. Because, if they have the CE or the FDA, so the notified body or the FDA, they have reviewed in detail the product, the technical file and technical documentation. Post-market, do the post-market as much as you can. After the product being placed on the market, you can make a market surveillance, you can make follow-up with the blood bank in order to check, to ensure that the product is performing as required, and then you follow the vigilance procedure. Now let me tabulate what is happening with each type of inspection. For the tightened inspection, with countries with tightened inspection, they are taking about from 18 months to 36 months until they make available the product. Reliance on other approval, they are not relying on other approval because they are relying on their expertise. Market availability of the product very low, sometimes no availability. So if the constrained resource countries follow this, so the product might not be available to save the people from virus inactivation. Resource required accredited laboratory. If you need to follow this, you have to have an accredited laboratory, trained expert committee, which need very robust system inside the national authority. Appropriateness for high-risk device, this is appropriate. Yes, I can agree that this might be appropriate to do. However, the product availability becomes very, very low, and it takes a lot of resources in the national authority to approve one device. However, the relaxed technique, less than 1 month, they can approve the device, and the reliance on because they rely a lot, and the market availability, you will find the devices are available. However, the resources required here is one trained inspector can do everything. So, however, this methodology is not appropriate, can only be used for low-risk device. And virus inactivation is not a low-risk device, so I do not recommend the relaxed inspection technique. Pragmatic inspection technique, this is a technique which enables the national regulatory authority to approve the device from 3 to 6 months, because they rely on other national authority approval or the FDA or CE certificate. Market availability, you will find the product available after maximum 6 months in the market. So here, one trained inspector is enough. So you have little resources available in the national authority to apply the approval of such a product, which I consider it appropriate as well. Conclusively, pathogen inactivation medical device is being used for decades, while no notable trend for complication associated. There too, the benefit of pathogen inactivation medical device outweighed the risk of the device. Pathogen inactivation medical device undergo several steps before being used directly to the patient. I mean here in the blood bank. And it is possible to be inspected 100% because, the people in blood bank, they can inspect the device before going to the transfusion process, which minimizes the risk and severity of any associated risk. Currently, viral infection become contingent and the control thereof become unintelligible, and need for virus inactivation become a prerequisite in each national regulatory authority. Pathogen inactivation medical device can be authorized using pragmatic inspection technique discussed before. National authority, local manufacturer of pathogen inactivation medical device who do not have an approval from other NRA or CE shall exhibit tightened inspection. So the national authority can approve their own, the manufacturer cited inside their countries without having approval by having the tightened control described in previous slides. In case of emergency situation approved by national regulatory authority, relaxed inspection technique can be utilized under special condition decided by the national regulatory authority. Finally, after the product being approved by the national regulatory authority, the blood bank, it is not only his rule is to use the product, but they have to use the product according to the instruction for use associated with the product. Prepare incoming inspection procedure to approve the pathogen inactivation product before use. If the product found to be non-compliant before use, send a complaint to the manufacturer. Report to the national regulatory authority if non-compliant product being used. Report the performance of pathogen inactivation product annually to national regulatory authority. By this function or this rule of the blood bank, now we consider that the national regulatory authority being informed or got enough feedback for their market surveillance. So the summary of this, the regulatory oversight for IVDs and a screening test kit used for donor selection, testing of blood and blood component has mostly been in place under the Minister of Health or National Regulatory Authority and the FDA responsibility, because hospital and the clinic use these products. However, regulatory oversight for medical device involved in preparation of blood component, e.g. apheresis machine and blood bags, and associated substances, e.g. anticoagulant and additives, additive solution is not yet established with some product. Only blood establishment use this product. Some technologies are even not in the process of preparing regulatory framework, e.g. apheresis system, PRT system, bacterial contamination detection system, etc. For this product, pre-market review and the approval relies mostly on the review and the approval from other international recognized bodies such as WHO, FDA, EU, MDR requirement, PEI assessment, etc. Thank you.
Hello, so we have now the time for the questions to the speaker of this session. But before we move to the Q&A, I would like to put things in context. Yesterday, we heard about various approaches like quarantine, technologies for pathogen reduction and viral inactivation in bag system for single donor or mini pool plasma, cryoprecipitate, or even immunoglobulin. And what the session of today is trying, and it's probably something which is a bit new, we are trying to think out of the box, is to try to identify whether or not it is realistic to go one step further in the pool size at domestic level, one step further in sophistication of the processing, until the volume of plasma or the volume of quality plasma which is generated at domestic level can meet the requirement for a next step, which could be contract fractionation domestically or at regional level. Whether or not there is a possibility to develop cost-effective, small-scale technical options to prepare products such as immunoglobulins or factor VIII, so that plasma is not wasted. This is what we want to avoid in this intermediate phase. And we also wanted to involve industry equipment supplier and see whether they can facilitate, help or not the implementation of such technologies, looking at regulatory implication, training and GMP requirements in low and mid-income countries. So having said this, we are going to move to the question, and we will take into consideration a few questions which have been asked in the Q&A. And I will direct the first question to Dr. Suma Ray, and I will have a similar question for Dr. Mariangela Vargas. So Dr. Suma Ray, you have presented what I believe is an impressive increase in the scale of production over the last few years, in a very short period of time. And you also presented briefly, but you presented the standalone production method, which was developed from the anti-SARS-CoV-2 immunoglobulin, using a process which is slightly different from your usual fractionation process, and it is based on caprylic acid precipitation, affinity chromatography, ion exchange chromatography, ultrafiltration, nanofiltration, giving a product with good level of purity and subjected to two dedicated steps of virus reduction. So having said that, my question would be, how do you think is the feasibility for another or for a low and middle income country to introduce such a technology at a relatively small scale, get the regulatory approval for such processes for the production of a normal immunoglobulin? So how feasible it is, how difficult it can be based on your experience?
Good day to all, and thank you, Dr. Thierry, for this question. Well, technically, yes, definitely it is feasible by all the countries, all the LMICs. Economically, I would have a varied opinion on the different these unit operations that you have mentioned. So with octanoic acid, I would say it is definitely feasible economically. With the virus filtration step, yes, it is a bit difficult, but then again, it's a trade-off between safety and COGS. So I think the balance should tilt towards the safety side. And in this context, I think even the suppliers can be supportive, just as you said just now, and have a different price strategy for the LMICs so that it is a win-win situation both for the plasma manufacturers, for the suppliers, and most importantly, the patients. Nevertheless, I think there cannot be any trade-off with safety. With regards to the affinity chromatography, yes, it is a difficult, very difficult proposition, and I wouldn't advise it for normal immunoglobulin where the volumes are much, much larger than the COVID hyperimmunoglobulin, which is similar to the manufacturing of the other hyperimmunes, which are in much smaller scales, where it is quite possible.
Okay, thank you very much. So I have a similar, before Dr. Micha Nuebling asked a question, I have a similar question to Dr. Mariangela Vargas. So don't forget to turn on your camera, your video for all the speakers. Mariangela, you have been showing that the producer of antivenom immunoglobulins are mostly located in low and middle income countries nowadays. And they are a demonstration that fractionated IgG for intravenous administration, which is the case for antivenoms, can be manufactured in low and middle income countries from relatively small pool of plasma. And in your presentation, you show that the process you use combined caprylic acid precipitation, additional step for Derived filtration, aseptic filling, which are all very similar, as you said, to what is required in the human for the human plasma fractionation industry to make human IgG. So again, taking your experience in Costa Rica at Instituto Clodomiro Picado, what do you think of the feasibility for low and middle income countries to implement such kind of technical approaches for human IgG, maybe with additional step for purification? So what do you think, Mariangela, how feasible it is? And also, since we have the question, maybe you can comment on the purity of the antivenom immunoglobulin which you are producing.
Thank you. Thank you all for the invitation. And thank you, Thierry, for asking this important question. Well, I think that it depends on a lot of factors. If it is going to be a actual fact that all the low and middle income countries that want to start in this pioneering objective of having PDMPs production in a domestic way, it depends on a lot of factors. But ones that I want to emphasize and not necessarily entering in the technical fact is that it's important to start with an approach that is mainly focused on the persons, the people, the group of people that is going to participate of all these initiatives, because people is the basis of any productive initiative. So it's really important to start with a focus in the human resource that is going to become part of this initiative. So saying that, all the philosophy and all the mission and common objective on this is once you can have that and a really good basis, then you can start thinking of the different issues, that the regulatory issues, the plasma supply, the technical gaps, the financial fact that is really, really important. But I think it is possible. As I said, most of the antivenom producers that supplied antivenoms to envenom patients, 70% of them are public institutions. And in this way, it is just to having it clear that it is a process. It cannot happen from one day to another. it should be a process that it needs to grow in a step-by-step, first at a small scale, and probably starting with IgG, that is the driver product in the plasma-derived industry. So it's really important to know that it is possible. We have done it at ICP, but we started since the middle of the past century. So it's a gradual process, and probably it's easier if we focus first on the human resource and also in cooperative ways to solve it. So probably it's easier if in a region you have several LMIC countries that want to participate on this initiative, and you can have a network of different small manufacturers that are working in this initiative, so that all the different things and issues that have to be assessed are done in a cooperative solidarity way. It's easier if you start that way, and not only to have all the facility and all the infrastructure and only thinking in that way. So I think it's possible, yeah.
Muchas gracias. So now I hand over the microphone to Micha Nuebling for questions.
Yeah, unfortunately, my video has been stopped by the organizers, but nevertheless, my voice is still there. And I wanted to ask a question to Dr. Gonim. You mentioned, or you explained, the regulatory approach in our countries for devices, and it's quite complex. Sometimes maybe it's too complex. And could you imagine at least that in other parts of the world, a more streamlined process could be used, which makes not so much compromise on quality or on potential, let's say, potential risks which come up, but in order to start such a regulatory process, to skip some of the elements and to put them in place at a later stage, because otherwise it could be overkill from the very beginning. This is my understanding, at least.
Thank you for the question. Actually, reliance is being used since ages, actually. So, for example, if we have a look to FDA, you will see that for the medium and low risk devices, they have a procedure like a 510K. And for high risk device, they are using the PMA pre-market approval, which is the same technique that they have offered. So whenever we have a product with high classification, it has to go through the very restricted methodology according to the FDA, like PMA. However, we have to relax some of the requirement to allow the product to be placed on the market to save people. But I'm speaking in my presentation specifically for a virus inactivation product. Virus inactivation product here, if it has been handled, the bags or the cascade separately rather than the chemical or the reagent, it can be handled in middle class devices. In this case, you will find that the product is lower classification, so it is not that high risk product, so which can be compromised about regarding the requirements, because think about the point from the other point of view. If we are handling, or as a competent authorities or national regulatory authorities, and we continue to review the product and reinvent the wheel and redo the work being conducted by notified body, however, we don't have enough resources. Here, the constrained country is not only constrained for money or for finance, it is also constrained regarding the competence of the people in order to break through this sophisticated methodology. So now I have the product, if they have been approved by FDA or CE, so there is an immense review of the product, and there is a specialist reviewing each part of the product.
So this can allow the competent authority or the national competent authorities to reduce and to allow the product to be introduced to the market.
Okay.
I hope this answered the question.
Yeah, it answers. It's interesting. Yeah, I see. I have another question to the ladies from Merck Millipore and also Sartorius. Are there products which you have shown here, are they CE marked, or are they mainly distributed in the Asian market or in low and middle income countries? So maybe
I can.
Yeah.
Thank you. So I think all the products I showed, so Jian-Fei and I showed in the presentation, are used worldwide for all kinds of pharmaceutical products. So that nothing is dedicated only for Asia so far. It's used in GMP productions.
Okay, thank you.
Yeah, I think same for us. Yeah, they're all marked for GMP production and they are used worldwide in Asia, Europe, US, yeah, everywhere. So yeah, if you have questions about any specific product, I can double check on those.
Yeah. Okay, thank you.
Okay, so maybe I can. I have also a few questions concerning to the three industry suppliers. I will follow the order with Sartorius and Merck Millipore, then Asahi Kasei. But we had several questions about the cost of single-use technology, and also we would like to have your opinion on the ease in your view, because you have a broad view of the plasma fractionation industry, high-income countries and low and middle income countries. But what is your opinion about the ease of implementation of plasma processing technologies in low and middle income countries? Because you have unique experience in this field, and you are also selling products for vaccine production or antivenom and other biological in LMIC. So how easy do you think it would be to implement new technology in low and middle income countries, and what can you comment anything about the cost of single-use processing or single-use technology? So starting with Prity Bengani-Lutz from Sartorius.
Sure, yeah. So I think single-use systems, they're definitely easiest to implement. You know, they can, they also, the good thing is that, you know, there's a low, can you guys hear me well? There's a low risk of contamination, right? There's no cleaning of the hardware involved, no validation, you know, lower, so you don't have to do like this time that's spent on validation for cleaning and hardware cleaning and things like that. You don't need that. Also, you know, you save on CapEx and it gives you the flexibility. More importantly, it gives you the flexibility, because when you're starting out, you know, you're trying to figure out basically your process and things like that and what scale you would need. And so if you buy expensive stainless steel hardware, it kind of you get locked into that size, and you know, but if you can delay that decision and kind of start off with single use, then, you know, you reduce the risk almost sixfold, because you get more data and, you know, more time to really figure out how much you need, and so you're not locked into it. So I think single use definitely gives you the flexibility, lower CapEx, less labor cost, less hardware validation requirements, no cleaning requirements. Yeah, so I think cost-wise, it definitely, I think in the beginning, the consumables may be a bit expensive, but I think but when you see all the benefits that you get, I think the benefits far outweigh the cost, and the payback is sooner.
Yeah, thank you, Prity. Yeah, same question to Josephine Cheng, as well as Jian-Fei Shou from Merck Millipore. I don't know who is going to talk.
Okay, I can.
Josephine, yeah.
So, thank you. I can talk and then Jian-Fei can comment. I think like Prity explained already all these benefits for single use, right? But for the cost wise, I think before it's not just maybe you see the spend in the consumables at the beginning, but we did actually run this biosoft modeling using the generic IgG as examples to show you in the presentation that where when you are having small scales to start for low and middle income countries, right, then you have a cheaper price actually comparing to all the capex investment for the standard steel systems. And when you have larger scale, then the differences become closer. Yeah, but I think to the level of flexibility or the ease of employee training, single use is definitely a way to start. And it is already a common trend in the vaccine industry. And the vaccine, you know, the selling price is much lower than the plasma-derived medicinal products. So we are quite comfortable that you can also utilize the benefits of single use in starting of your domestic production. Maybe it's
okay, thank you. Yeah, okay, thank you, Josephine. Yeah. And now we can ask the question to Dr. Tomoko Hongo from Asahi Kasei Medical. And there was also a question about nanofiltration, whether it is an independently run system in a manufacturing process. And I think probably you will say, yes, it's something which is independent and run independently within or inside a manufacturing process. But Tomoko Hongo, do you want to comment about the single use and advantage for the industry in low and middle income countries and feasibility?
Yes, thank you for your question. So in my consideration, our filter is a single use and also autoclave ready to use or pre-wetting, so not necessary to a pre-cleaning system. So easy to connect use to the ready to start with the filtration. So they're easy to operate. So we can support the training operation easily. So a scalable filter for single user system. So the easy to straightforward to the scalability. So I think so. This is our merit. Okay, this is my answer.
Okay, thank you very much. So I think that considering the time, I would like to thank Dr. Micha Nuebling, the co-moderator of this session, as well as the speakers for the preparation of their presentation and the answer to the questions.